So 12th class, in the last class we had discussed that what is the use of plasmid, how does it act as the vector. So today I am going to discuss that what are the different tools that are used in this recombinant DNA technology. So before starting the tools, there are many tools that are used in the recombinant DNA technology. We will use that one by one. So first of the tools that is used in the recombinant DNA technology is the restriction enzyme. Now what is the meaning of restriction enzyme? Restriction enzymes, these are also known as molecular scissors. What is the meaning of scissors? Scissors means this is the, you can say this is the device that can cut a particular thing. So these are known as molecular scissors. These are known as molecular scissors, means these are used to cut. So when in the biology we say that we have to cut a particular thing, so obviously we will use there the restriction enzymes. So as in the last class we had discussed that the plasmid will be cutted with the restriction enzyme so that the alien piece of DNA can be introduced there. The restriction enzymes are used to cut the things, right? Now these restriction enzymes, what are actually these restriction enzymes? These are the specialized enzymes that recognize and cut a particular sequence of DNA. For example, once again I am drawing so that you can easily understand the concept. This is the plasmid and we will cut it from here like this. So this cutting for the insertion of alien piece of DNA here, this cutting will be done by the restriction enzymes because these are the molecular scissors. Now these restriction enzymes are of two types. One is the endonucleases and second is the exonucleases. As the word itself indicates, exo means exit, external, endo means internal. So if I say that this is a particular piece of the DNA, suppose this is a piece of DNA, and I am also drawing one more. This is also a piece of DNA. Suppose this is the first one and this is the second one. And if I say that in one case the enzyme act at the end and in one enzyme act as the middle. Now this is the difference between these two. One type of enzyme will act at the end, means on the external side, and second type of enzyme will act at the middle, means in the internal sequences. So if an enzyme is acting in the middle, you can say it cut the DNA at specific position within the DNA. Within the DNA means this is within the DNA. This is showing within DNA, means not on the ends. So if the restriction enzyme is cutting a particular segment in the middle, that is known as endonucleases. Exonucleases, you can see if it is cutting the DNA at the end, in this you can see that the enzyme is acting at the end. It may act at this end, it may act on this end. So if the enzyme, they are cutting the DNA from the end, then you will say these are the exonucleases, and if the enzyme is cutting the DNA from the middle, then you will say it is the endonucleases. So these are the basic differences between these two types of the enzyme, that is exonuclease and the endonuclease. But there is quite few interesting features regarding the working of these restriction enzymes. Now first feature, every restriction enzyme is having its own recognition site. Every restriction enzyme is having its own recognition site, and till now approximately 900 restriction enzymes have been isolated. Second, this was the first interesting feature of these restriction enzyme. Second point regarding the working of these restriction enzyme is that the sequences which are identified by these restriction enzymes are known as palindromic. Now, what is the meaning of this palindromic? These are the sequences of the base pair that will be same both backward and forward. For example, if you study this sequence, so how will you read it? You will read it like G, A, T, T, C. This is the sequence that you will read from 5' to 3' direction. Now in the second one, in the same direction, if you read from 5 dash to 3 dash, what will be the sequence? G A T C T T C. If you read in 5 to 3 direction, if in the first it will be GAATTC, in the second GAATTC. Now what will you observe here? You will observe that whether we are on this strand, this is the 5-3- strand and this is the 3-5- strand, but if we read the direction in the same sequence, means if we are reading from 5 dash to 3 dash, then sequence will be same. Now if the sequence is same, that is known as the palindromic sequence. Each and every restriction enzyme is having its own particular palindromic sequence. You can easily understand it with help of a word Malayalam. If you are reading this word either in any direction, you will always get the word Malayalam, whether you are reading from the first time or you are reading from the last time. So these are the features of the restriction enzyme. Now the restriction enzymes, it doesn't exactly act on the palindromic. It acts a little away from the palindromic sequence. You can see it cut a little away from the center of the palindrome. This is the center, this is the center, this is the center, but now you see the restriction enzyme is not working at the center, they are working a little away. When these restriction enzyme will act on the sequences, these are attacking between the G and A, so obviously G and the remaining part will separate. Similarly here, this is acting between G and A, so G and remaining part will separate. As a result of separation of these G, these are known as the sticky end. The sticky end will get exposed. All these processes form the basis of the RDT, that is the recombinant DNA technology. Now if this is the one part and this is the second part, this is the one part which is formed due to the action of the restriction enzyme. Now these two will get joined, and that joining is because of the enzyme known as DNA ligases. So it means that if we cut a piece of DNA, so which enzyme will be used there? Restriction enzyme. And if we want to join these two things, then we will use the DNA ligase there. So the use of these two enzymes must be very clear to you, that if you want to join two strands, then you will use DNA ligase. If you want to separate, if you want to cut a piece of DNA, then you will use which enzyme? You will use restriction enzymes.